So, hello, everyone. Thank you for joining us today. My name is Javier Brum, and I'm leading the organization committee for this year's IEEE-UFFC Latin American webinars, together with Patricia Cardoso de Andrade, Andrea Pulido, Sophie Morse, and Colin Brick from Conference Catalyst. It is our pleasure to kickstart this second webinar of this Latin American series. So before starting, I would like to make some announcements that may be of interest to the ultrasonic community. So this year's IEEE IUS conference will happen in a hybrid format. So you can either fly to Venice or participate from your PC. Early registration will end on September 5. So please register to access all sessions and panels. In addition, and very important, this year we will have for the first time an LIUS Latin American ultrasound panel at IUS, where we'll be discussing the global opportunities and scientific challenges for ultrasound-based research in LATAM. So please visit the conference website for more information. So, and here's an job opportunity at Verasonics. Verasonics is hiring a full-time ultrasound scientist based in Medellin, Colombia. So please, for more information, check verasonics.com or contact directly Dr. Miguel Bernal. So a few details on today's seminar. So as usual, you can type questions by using the chat function anytime during or after the lecture to be addressed in the Q&A session at the end. Questions can be in Portuguese or Spanish if preferred. Andrea and Patricia will be handling translations. The entire session will be recorded and placed on the Society's YouTube channel for review or for those who are not able to join us today. So today, we will have an early career speaker from Mexico and a senior speaker, from Chile, now at Switzerland, and a senior speaker from Brazil. As a moderator for this second webinar, we will have Dr. Karen Folke Sepúlveda from Mexico. She is an associate professor at the Institute of Physics of the Universidad Nacional Autónoma de México, UNAM, where she leads the Optical Micromanipulation Laboratory. She's an expert in optical and acoustical trapping, topological and dynamical properties of structured wave fields, analogies among different wave systems, and nonlinear optical phenomena in colloidal media. So thank you, Karen, for joining us here today. And of course, thank you very much for accepting moderating this webinar. So please, I will stop share the screen and please go ahead, Karen.
Thank you very much, Javier. Thank you all for the organization of this webinar and the audience. I also want to thank all of you who join us today. Well, I have the pleasure to introduce first Héctor Estrada, who will give us a talk about optical-guided transcranial ultrasound stimulation in mice. Héctor Estrada studied acoustical engineering in Chile and obtained his PhD on physical acoustics from the Universidad Politécnica de Valencia, Spain, in 2011, investigating the ultrasound propagation through photonic plates. In 2012, he joined the Institute for Biological and Medical Imaging, Helmholtz Zentrum, München, as a postdoctoral fellow developing hybrid optoacoustic ultrasound neuroimaging system and studying the acoustic properties of the skull using lasers. He currently works at the university and ATH Zurich developing models and techniques for transcranial ultrasound and optoacoustics. His research interests include optoacoustic and ultrasound imaging, guided ultrasonic wave propagation in bone, and ultrasound neuromodulation. Thank you very much. We will see the video of Héctor's talk. Thank you.
Hi, everyone. Thanks for joining this talk. Thanks also to the organizers for inviting me. And I would also like to thank my co-authors and collaborators that make this work possible, and also thanks to our funding agencies. Today, I'm going to talk to you about how we can use optical techniques to guide transcranial ultrasound stimulation in mice. I assume you know already that mechanical waves like ultrasound can affect neuron activity. So if you turn on the ultrasound on a neuron, you will see, for instance, this calcium change here when you use fluorescence for imaging, and if you stick a needle, you will record this electrical signal that shows the neuron firing after the stimulation using ultrasound. How this happens? Well, it's still unknown. There are a few things that might be going on, but there is no consensus yet. This could be temperature, could be also bubble formation and cavitation, also in between the lipid membrane that covers the neuron, or it could just be the response of ion channels to mechanical force. So it's actually very challenging. And if it's challenging, why anyone will be interested in solving this issue? Why would anyone be interested in knowing how ultrasound changes neuronal activity? Well, the reason is that ultrasound has very high potential because it's non-invasive. So it can be non-invasively used through the skull for therapy. And we are talking about here low pressures, not like the ones that are used for high flow therapy, where you cook tissue inside of the brain. No, much lower, another regime. And because of this, is very promising. It has been proposed to help with epilepsy, Alzheimer, Parkinson, depression, and many other things. So there is a high potential here. But the field is also a bit of a mess because usually when you develop a therapy, you have first in vitro studies on cell cultures, then you go to preclinical studies, and then in mouse models or other animal models, and then at the end you go and you reach the clinical trial phase where you perform this and test this therapy in humans. But because it's ultrasound and at low pressure is known to be safe, what happens here is that you have simultaneously in vitro, preclinical, and clinical studies being performed. So it's a very messy field, but it has a very high potential as well. And in the realm of preclinical studies, the mouse brain, of course, you know, is very small. And if you use a nearly unfocused transducer with low frequency, you will just cover the whole hemisphere. This is not very helpful to do any experiments in mice. You can increase the frequency, for instance, and get a better focus, like this linear array. But what you really need, if you want a very high spatial specificity, is to cover a wide range of angles. And this is exactly what we do. At 3 megahertz, you can get an even smaller focus than this 5 megahertz focus here. But now the challenge is, yeah, I told you about using optical methods, but bulky ultrasound transducers are very opaque. How can you do actually any imaging there? So our solution is to use an aperture that is here in the spherical array. And through this, we can just cover and use wide field fluorescence through the whole mouse cortex, as you can see in this image. So it's actually very neat because it's a spherical transducer. We can use three-dimensional beam steering. The skull at 3 megahertz, which is the frequency we're using, is not a big problem. Only 6 dB decrease in the pressure, and the focusing is still very nice, below 400 microns. And what we observe here is surprisingly a fluorescent dip when you use a pressure of around 3 megapascals. So there is this dark spot that indicates that there is a negative change in fluorescence. If you take a look at the time trace, at this particular point, you will observe something like this. So you have the calcium transients because of the resting state of the mouse brain. So when the mouse is anesthetized, you have a very synchronous activity that is called resting state. And only when we turn on the ultrasound during 150 milliseconds, you get this decrease in fluorescence that only lasts till we have the ultrasound on. We turn it off and it goes up again. We had to develop some methods to clean the signal, because the resting state, as you can see, is very, very large, because all the neurons are working more or less synchronously. And we actually use beam steering, and we observed that this dip actually follow the position of the transducer focus in the mouse brain. You can see here we target the red spot. You can see here the dip, and we move the target to this green spot, and you can see the dip happening 10 seconds after. So is this dip related to neurons? Actually not. We perform exactly the same experiment on bacteria and observe exactly the same. So it has to be something more fundamental, and in fact it's just a thermal effect. So when you have fluorescence and you increase the temperature, the fluorescence brightness will decrease. As you can see here, we tested this in a slide of a mouse. I forgot to mention, these are transgenic mice, GCAM6F. They have fluorescent neurons that increase the fluorescence when the neuron is firing due to the calcium influx inside of the neuron. And we fit this thermal model, and it's a spatiotemporal model, and the fitting is quite good, I would say, particularly if you take into account that this is all done in vivo through the skull. One drawback, though, is that we cannot measure absolute changes in temperature. We only have access to relative, and even then we have to perform calibration, but even in this case is very useful. So we can use this tag to know where in the mouse brain we are stimulating. But we also can do some other techniques, for instance, functional connectivity analysis, which is something that looks for the correlation between the different regions in the mouse brain. You can see here from neuroscience, we already know how the mouse brain is split into different regions that have different functions, and we can just correlate the time traces among them. This is an example for a baseline, and this is what we see with 3 megapascals. And after calculating the correlation, we can build this matrix where you have every region of the brain with every other. And for the baseline, it looks like this. And after 20 sonication with 3 megapascals, we don't see a clear change. Only after one sonication with 3.9 megapascal, we can observe that there is a difference. And actually, we perform histology. We couldn't find any signs of damage with the 20 sonications at 3 megapascals, but we observe a very small lesion, though, with one sonication at 3.9 megapascals. It looks like a burst vessel, very tiny vessel, as you can see in the image. So cool. So using calcium changes and functional connectivity analysis, we can spot when the ultrasound is inducing actually a change in the inner workings of the brain. And if we increase the pressure even further, we can trigger what is called cortical spreading depression, which is a biochemical imbalance that travels through the cortex. It's confined to just one hemisphere of the brain. As you can see here, it only propagates here. You can see the time traces. It's a huge change of fluorescence because it happens to all the neurons. And it's a very dramatic thing. It's not bad for the neurons outside of the region where we triggered the small lesion. We couldn't observe any of this with 3 megapascals, but if you increase it at 4.5 megapascals, you see and you trigger a CSD every single time. So yeah, I hope I convinced you that the fluoro-thermal tag can be very useful to report where the ultrasound focus is in the mouse brain. but it can also be useful to spot thermal confounds. So if you are using fluorescence for your measurements and you want a clean signal only coming from the ultrasound, if you see this decrease in the fluorescence, you can be sure you have a thermal effect. So you're increasing the temperature of the brain locally with your ultrasound, and then you have to account for that in your analysis. I hope I also convince you that this method and FLUS is a valuable tool to assess ultrasound safety. You can see what happens in the brain. Some other experimental paradigms don't have access to this, and I think this is actually a need if you want to assess how safe ultrasound will be when delivered to the brain. And I hope I convince you that this tool and our methods have a high potential for studying ultrasound neuromodulation.
Well, we thank Héctor very much for this wonderful video talk. And now I have the pleasure to introduce Marco Antonio Brizotti Andrade with the talk Acoustic Levitation Methods for Suspending and Manipulating Objects in Midair. Marco Andrade is a lecturer at the Institute of Physics of the University of Sao Paulo, Brazil, and his main research interest is to develop new strategies to levitate and manipulate objects with sound. He received a bachelor's degree in physics from the Institute of Physics, University of Sao Paulo, Brazil in 2004, and a master and doctoral degrees in mechanical engineering from Polytechnic School, the University of Sao Paulo, in 2006 and 2010, respectively. Well, now I leave you with the talk of Marco, and at the end, we open to questions. I remind you that you can ask at any time in the chat, okay? Thank you.
Hello. First, I'd like to thank you for the invitation to give this talk, and I will talk about the acoustic levitation methods for suspending and manipulating objects in mid-air. Basically, this presentation will be divided in the following topics. First, I will give a brief theory behind the theory used for suspending objects using sound wave, and then I will present 4 different acoustic methods for levitating objects. These methods include the standing wave, inverted near field, the far field acoustic levitation, and the single beam. So let's start with the theory. The idea here, suppose that we have an object, this one, and then we have an incident wave that is described by the incident pressure and the incident particle velocity. When this wave is reached the object, then we will have a scattered wave. And if we add both waves together, then we have the total pressure and the total particle velocity. And if we want to calculate the force that this wave will apply to the object, then we can use this equation that basically it calculates the acoustic radiation pressure that acts in the object surface. Then if we integrate the acoustic radiation pressure over the object surface, then we have the acoustic radiation force. For the specific case, for an object whose size is much smaller than the acoustic wavelength, here we have a spherical object of radius r, that's much lower than the wavelength, then we can use the Gorkov equation. What's the idea? This Gorkov equation says that if you have the incident pressure and the incident velocity, and then use this, we can use this equation for calculating the Gorkov potential that's represented by this letter U. Then this is a gradient potential, so if we take the gradient of the potential, then we have the acoustic radiation force that acts on this small particle. The advantage of using the Gorkov equation that we don't need to take the scattered wave. It only if you have the incident wave that reaches a certain particle, then we can use this equation for calculating the potential. So this is the theory. Now let me introduce the acoustic levitation methods. Basically, we can divide the acoustic levitation method in 5 types. We have the standing wave method that suppose that we have here a transducer and then a reflector, and this transducer is vibrating at an ultrasonic frequency. Depending on the distance between the transducer and the reflector, we will have a standing wave, and then if you insert a particle whose size is much smaller than the wavelength, then we can trap this particle at the pressure node of the standing wave. So this is the standing wave method. We can also have the near field acoustic levitation, where a flat object can be levitated slightly above a transducer that vibrates at an ultrasonic frequency. So here we have a very small gap between the object and the transducer. We can also have this other type of levitation called inverted near field, that here we have the transducer and the object levitates below the transducer. Then these two methods are similar, but depending on the transducer radius, we can have or the near field or the inverted near field. Then we can have this other method that we call the far field levitation method, and here instead of using a reflector, we have the transducer and the object levitates at a certain distance from the transducer, and here we have a standing wave between the object and the transducer. And finally, we have the single beam acoustic levitation methods. With this method, we don't need a reflector. So basically, we have a transducer or an array of transducers, and then we need to generate a specific pressure field distribution to suspend this object. So this is the main idea, and now I will comment about the standing wave method, the inverted near field, the far field, and single beam. This one I will not talk about this method in this presentation. So let's start with the standing wave acoustic levitation method. So the idea here, as I said, suppose that I have a sound emitter, and here we have a very small object, and below we have a reflector. If the distance between the emitter and the reflector is approximately half the wavelength, and then if we turn on the emitter, then we will generate a standing wave, and then this object will levitate at the pressure node of the standing wave field. We can use the Gorkov equation for calculating the radiation force that acts on the particle. Here, instead of having the force, I have the potential distribution. So what we have done here, we have used the finite element method for predicting the pressure field distribution and the particle velocity distribution between the transducer and the reflector. Then by using the Gorkov equation, we will have this potential distribution, and here we have 3 positions of minimum potential. So if we place a particle closer to this position, then the particle will be levitated here. So here's a comparison with the experiment. Unfortunately, we cannot see this other particle that is around here. So here's a small video. We can use this method for suspending not only solid objects but also liquid droplets, as we can see here. And one potential application for that is to combine this kind of experiment with synchrotron radiation. Here we have a synchrotron facility in Campinas in Brazil. And so what happens here, this is a picture of the experiment that was done in this line. So here we have the X-ray that reaches the levitating particle close to here. When this X-ray reaches the particle will scatter the X-ray, and then by using an X-ray detector we can measure what's going on with the levitating particle. Here is an example of the data that can be extracted with this kind of experiment. Another possibility of using the standing wave method is to create a multifocal point acoustic levitator. So instead of using only one transducer, we can use an array of the transducer. In this example, we have an array of 16 by 16 transducer that is divided in two groups. One group is represented by the blue color, so we have here the blue transducer and the red transducer. They are distributed in a chess pattern. So if we take the blue transducer and define a focal point, and then we set the phase of this group of transducers such that all the emitted waves reach this point, this focal point, with the same phase, then we will have a constructive interference here, and the superposition between the incident and the reflected wave will generate the standing wave, where we can trap small objects in the pressure node of this standing wave. And then by changing the phase, we can move this standing wave. If we have inlets to insert the droplet, then we can levitate these droplets at the pressure node of the standing wave. You can do the same with this red group of transducers, and then by controlling the phase we can levitate two droplets and then merge them together, and then after doing some kind of chemical reaction, for example between the two droplets, we can move the merged droplet to this outlet, and then by turning off the transducer the droplet will fall out in this outlet because of the gravity. So this is the concept. Based on this, we have designed an experiment to perform droplet manipulation and combining them together. We have also designed a piezoelectric device for injecting droplets into the levitator. So here we have a liquid reservoir that's attached to this device, and then by applying an electric pulse to the piezoelectric material, then it will inject a droplet here into the devices. So we have used this array here on top of our levitator. This array is described in this paper. So here we have 256 transducers emitting at 40 kilohertz, and each transducer has a diameter of approximately 9.8 millimeters. So here we have the simulation. Here we have the red transducers focusing at this point, and this is the acoustic pressure distribution over time. So the blue transducer for this particular case are not working, but we can set the phase of this blue transducer to focus at this point located at this position, and by doing that then we have these two standing waves, and then by controlling the phase we can move these waves horizontally. So here's the acoustic pressure amplitude, and this is the Gorkov potential, the simulated one. But okay, this is a static figure. Let me show the video. To record the video we have used both a normal camera and also a high-speed camera to observe the droplet motion. So Here on the right we have a picture of the experiment, the array of transducer on top, here the reflector, and below the reflector there is the droplet injector. Let me show you now a video of this experiment. Here we have the simulated Gorkov potential, where we should levitate droplets in this position of minimum potential. So let me run the video, and here a video of the experiment. The video here was recorded in real time first, so the droplet was injected into the levitator using the inlet on the right. Then, after waiting a few seconds in order to stabilize the droplet, then we start to change the phase of the array such that we can manipulate the droplets. After that, we will inject another droplet into the system, and then by manipulating the phases of both groups of transducer, then we can merge the droplets to make, for example, chemical reaction. And then after that we can move the droplet to the outlet, this small hole located here, and then by turning off the transducer, gravity will do its job and the particle will fall out. Okay, this was recorded with a normal camera. Let me show you now a video recorded with a high-speed camera. So this is much more interesting. So here we have the droplet injection. So after the droplet is injected, it will be levitating at the pressure node of the sending wave. Then we also have the merging of two droplets. That is shown here. Yeah, the dynamics of the interface is very interesting, as we can see. And then after merging the droplets and moving it to the outlet, then by turning off the transducer, the droplet falls here because of the gravity. So one possibility for this kind of device is to conduct chemical reaction without using test tubes. And then we can also automate everything. Okay, but now let me show you another kind of levitation that we call it inverted near-field acoustic levitation. What's the idea? First, if you have the near-field levitation, where we have an object levitating above the transducer, if we inverted all the device, all the setup, then we have this kind of levitation that we call inverted near field. So here we have a picture of an object being levitated by this technique. In order to understand how this technique works, we have also implemented a finite element method using an axisymmetric model. So here we have the transducer vibrating with a velocity u0. Here we have the object that can also be a reflector with the same radius as the transducer, and then we have this distance h between the transducer and the object. And here we have the air medium. So if we simulate the acoustic pressure and the particle velocity between the transducer and the object, then we can calculate the radiation pressure using this equation, and by integrating the radiation pressure of the object's surface, then we have the force. So here, instead of using the real quantities, we prefer to work with the dimensionless quantities. So the dimensionless radiation pressure, dimensionless force, where this area C0 is basically the radius, the area of the objects, P multiplied by the R squared. So by performing this simulation for different values of radius and this this distance, then we can create this kind of result where we have the force as a function of weight divided by lambda and the radius divided by lambda. And here it's very interesting to analyze this figure, because in this region for radius divided by lambda above around 0.4, not exactly 0.4, but in this region we have a repulsive force between the transducer and object, and in this region we have an attractive force. So if you plot here an example, in this dashed line we have a repulsive force. The force will be like this, and this black line that was obtained by R divided by lambda equals to 0.3, you will have this kind of force that is negative, so attractive force. So based on this result, we can say that the condition to obtain the repulsive force is this one, R divided by lambda higher than 0.38, and for this case we have the near field levitation. And if this condition is satisfied, then we have the inverted near field acoustic levitation, where we can levitate objects below the transducer. So based on this, we have designed a device for levitating and manipulating SMG components. So we have designed a device to levitate this particular component size. Then we have designed a Langevin transducer that operates, I don't remember the frequency, but I think it was 21 kilohertz, where we can levitate the objects below the transducer chip. Here is a picture of the levitating SMG component. And here we have a video of the device in operation. So we can use this device for manipulating objects. We have tested this with SMD components, but this could be useful for manipulating very fragile objects, or objects that can be damaged if there is a contact between the gripper and the component. So with this kind of device we don't have this problem. Now let's introduce another type of levitation that we call the far field acoustic levitation method. Here basically the method, we have the emitter, and then instead of using the reflector, because here if we have this condition, this standing wave will apply a radiation force to the reflector. So if we replace the reflector by the object, then we can suspend the objects using this approach. So here we have used the 3 different Langevin transducers to levitate this ball of 50 millimeters in diameter. That is much larger than the acoustic wavelengths. So this is a picture of the experiment, and here we have a small video where we are inserting the sphere into the levitator. We can also have a variation of this technique. Instead of using different transducer, we have designed this device. We have one Langevin transducer here, another one attached to an aluminum plate. And when both transducers are operating at the same frequency, then this aluminum plate will vibrate like this in its flexural mode. This vibration will also generate a standing wave between the aluminum plate and this object that you can use for levitation. It's interesting here because we also have lateral stability. So this is a picture of the experiment, and here we have a small video showing this levitation in operation. So yeah, this is the object. There's no wires to suspend this, so it's a real levitation. So this is the far field acoustic levitation method. Finally, we can also have the single beam acoustic levitation method. That basically what we have here, we have an array of the transducer where we can set the phase of each transducer independently, and the idea is to determine what are the phases to generate a specific pressure field distribution to suspend this object in the air medium. So we have done one possible strategy was done here. So here we have a mesosphere. I think it was, I don't remember its size, but I think it was around 90 millimeters in diameter. And by setting the phases of each transducer, then we could suspend this object in midair. We also have used this experimental setup for measuring the pressure field distribution. So here we have the comparison between the simulated pressure that's shown here and the measured pressure using a calibrated microphone. So this is how the pressure is distributed around the object. The object is levitating in this point. So the z-axis is pointing upwards. So these two figures shows the pressure distribution over the xy plane, this one the zx plane, and this one zy plane. So as you can see here, the object levitates like here. This is a isosurface of the pressure distribution. So here we have the object and the pressure distribution. We have similar to having like this. So this is our pressure distribution. So that's it. As a summary, I have presented 4 different types of levitation. In this talk, I didn't talk anything about the near field. So here we have the standing wave, where we have a transducer or a reflector, and we generate a standing wave between both, such that we can suspend a small particle into the pressure node. Here we have the inverted near field, here the far field, and this one the single beam acoustic levitation. Thank you so much for your attention, and I am open for questions.
Thank you very much, Marco, for a wonderful video talk. We have several questions. I will read the questions. I'm sorry, it's sorry, I have a technical problem here with the. Okay. First, we have a question for Héctor from Sophie. Héctor, for this technology to be useful for, say, Alzheimer's, is the idea that one would want to cover the entire brain? If yes, how would you do this?
Yeah, so first, some disclaimer. The setup that I showed is specifically fine-tuned to work in mice. So in principle, we can even use holography with this spherically focused array. It will be a bit less efficient, but we can do the beam steering and move the array if necessary. But in order to target humans, that's a completely different scale. And as I mentioned in my talk, there are, for instance, there are companies like Storz Medical that actually try some shockwave treatment for Alzheimer's. They didn't try mice, they didn't try in vitro, they just try in patients directly. They saw some improvement, but they have no clue how this happened. So I think the question will be to check in preclinical models where we have something like transgenic mice that have fluorescent neurons, something that we don't have in humans, to answer these more basic science questions. And if we want to target Alzheimer's in mice, I would say that in our group, we recently published a paper observing plaques related to Alzheimer using different method, optoacoustics and high resolution fluorescence. And I think there are many opportunities there, but specifically we will, if we want to irradiate the whole brain in one shot, we will need to change the device completely without giving up the imaging, which is actually very useful to understand what happens in the mouse brain.
Okay, thank you very much. Another question for you, Héctor. Can you tell me, this is from Marianne, can you tell me more about the device which emits ultrasound wave? Is it in vivo or in vitro?
Yeah, so it's designed to be used in vivo. So we perform routinely experiments in mice and actually also in awake mice with the head restrained. So it's designed to be used in vivo. But as I mentioned in my talk as well, to actually figure out what was this decrease in fluorescence that we observe in our experiments, we also use bacteria. So it can be used in both, but I would say is finely tuned for in vivo experiments. And if you want to use, for instance, a high resolution microscope combined with our system, it is possible, but it's actually also challenging. So we, particularly if you want to hold the bacteria or your cells or something like this, without having any glass interface, because as you might know, one of the challenges in ultrasonic neuromodulation to understand this in the microscopic level with a few neurons is that usually biologists will use a glass slide and then put the ultrasound, and this completely distorts, as as Marco shown, when you have a strong reflector, your ultrasound field changes completely, so it's hard to make the connection between a measurement performed in vitro with glass slide and what happens in vivo where you don't have any such structure in the mouse brain. So yeah, we use it in vivo and it could be used in vitro as well.
Okay, thank you. We have a question for Marco from Javier. What software did you use for the simulations?
For simulation, I think you made this question for the specific case of the array. For the array, we have implemented a code in MATLAB. So we use an analytical equation for a circular transducer emitting in the far field. Then what we do is basically add the contributions of every transducer. But for the other types of simulation, we use the console multiphysics. But for the array, it was MATLAB.
Okay, thank you. Another question for you from Kirti. Marco, how were you able to calibrate the sonic surface board to ensure the transducers were emitting in phase?
This is a good question, because when we buy the transducers, the phases are not, doesn't match, and there are variations in the phase, so we need to calibrate them. Basically, what we did for every transducer, we put another transducer to act as a receptor, and then manually we change the phase of the emitter until the phase coincides with a reference signal. So we do this for every transducer, and then after that, the array is calibrated.
Okay, thank you. Approximately, what was the volume of these droplets? Have you found limits on the volume and density?
Okay, the volume, I have no idea, but the ratio, the droplet diameter is around 1 or 2 millimeters. So we can change between, the typical the typical diameter is between 1 to up to 3 millimeters. It can be a little more, but not too much. And yeah, the typical limit is around 3 millimeters in diameter.
regarding the density, I don't know, because for liquids we have tried only to levitate water droplets, but with solids we can levitate up to 7,000 kilograms per cubic meter. But for liquids, most of the time we levitate water droplets.
Okay. Marco, what was the acoustic power and peak pressure that was generated with the transducer array of 40 kilohertz?
Okay, the acoustic power, I don't know, but the peak pressure, the pressure amplitude is around 3 kilopascals at the focal point, but it can be more, but usually it's around 3 kilopascals.
Okay. Is there an optimal droplet size to be manipulated with your levitation phased array?
Normally between 1 and 2 millimeters in diameter are the best diameters for us. Can be smaller and slightly larger than 2 millimeters, but in the range between 1 and 2 is the best.
Okay. Héctor, what kind of signals did you use for the functional analysis?
Yeah, so for the functional analysis, we basically acquire images, a video of the calcium signals changing over time. And these waves that are in the brain of the anesthetized mouse actually are very peculiar. They have very specific patterns, but the analysis we did was very simple. So we just pick a pixel in the image, and through the whole stimulation range, I think it's the recording, it takes a few minutes at 20 hertz, and we just use Pearson correlation. So we don't use any function to match, let's say, the response of the stimulation or of the brain. It's just a simple Pearson correlation between different groups of pixels. Although, for instance, we are using the same method, I mean, it hasn't been published yet, but with Optacoustic recording. And there we are actually using some, because with Optacoustic, you mostly observe vasculature, you see the blood very clearly. So we are using there some standard functions for hemodynamic changes. So then hemodynamic transients, yeah. So that will be mostly the difference. But for fluorescence, we are not using any specific function. It's just Pearson correlation between pixels.
Just one more question, because regarding this point, sorry to interrupt, but so what functional analysis shows you in this case was that the brain was damaged, that the loss of connectivity was due to a damaged brain or a damaged vessel in this case. Is that correct?
Exactly, yeah.
Okay, thank you.
Okay, so we don't have time for more questions. Javier? Sorry, okay.
Okay.
Sorry, do we have time for one more question or not even? Not even.
I don't know. Colleen, do we have time for?
Yes, we have time. We have 6 minutes remaining, so we have time just for a couple more questions.
Okay, I'm sorry, I am not able to make my own, but there are lots of questions, and that's good. Héctor, would changing the ultrasound frequency make a big difference to the bioeffects you would get?
That's the million-dollar question. It's very interesting. I think it makes a difference. And actually, from our own results, that, I mean, we are still processing the data, is not published yet. We have found a few surprises. I think the frequency plays a big role, but only if the changes are very substantial, like 1 octave. if you use similar frequencies, you tend to obtain very similar results. So I would say it makes a large difference, but only if you change the frequency a lot, which is the case, for instance, when you do experiments in mice with high precision, as in our case, or when you do something in humans with frequencies below 0.5 megahertz. So I think there is a difference there. And actually, that's a very interesting point that we will need to address in order to bridge this gap between preclinical studies and clinical studies.
Okay. Do you have a coupling medium between the mice head and the transducers, by the way?
Yes, it's degas water. We just degas water, and then there is a membrane, a plastic membrane, which is transparent to both ultrasound and light. Like that sounds very fancy. It's just normal kitchen foil.
Okay. Okay, thank you. Marco, have you thought in performing similar experiments for guiding gas bubbles in liquid media?
Yes, we have tried, but a very quick experiment. At the moment, I prefer to focus in air and then make all the experiments involving liquid media. I usually work only in the theoretical part, and my collaborators work in this, because the experimental setup, although the theory is very similar, but the experimental setup is different. We need the microscope, amplifiers work in a much higher frequencies. So, but yes, it's possible to guide gas bubbles. But here, in our lab, we prefer to focus only in experiments in the air medium. But yes, it's possible.
Okay. And do you have any comments on the transmit power needed to make those liquid particles levitate?
Yeah. Regarding the acoustic power, I don't know. I need to calculate from the pressure distribution. But in terms of the electric power that we apply to the transducers, it's less than 10 watts. So less than 10 watt. 10 watts electrical power is enough for suspending liquids and droplets.
Okay. And well, I will make a last question. We don't have any more in the chat.
Okay.
Well, it seems that you have a sort of stability issue with the droplets. They start to vibrate. And I'm wondering if this has to do with the size, and if you droplet generator, what's the reproducibility of the generator in terms of droplet size?
Okay. This kind of oscillation, what happens when we use the droplet injector to insert the droplet into the pressure node, it has an initial velocity. So if we wait like 30 seconds, then the oscillation stops. But of course, there are very small oscillations after, but the problem was that the droplet injector was inserting the droplet with a certain velocity, then it oscillates for like 30 seconds, and then it stops oscillating. Of course, there are very small oscillations, but
yeah, okay. Thank you very much. Well, Colin put in the chat a survey for today's talks. Please visit the page and complete the survey. And I guess we have no more time. And so we thank you all very much, and the speakers and the audience. And thank you all for your participation in the questions. And I give the word to Javier. I'd like to know if he has some final announcement to do.
Thank you. Hi, thank you, Karen. I couldn't find the unmute button. Do you see my screen?
We see your screen now.
Yeah.
We see, yeah.
Thank you. So just one final announcement before closing up this webinar. So thank you all for joining us for this second webinar. Thank you, Héctor and Marco, for these wonderful talks, and Karen for moderating this very interesting Q&A session. So our next webinar will take place on the 27th of October, featuring a talk by Stacia Marie Fletcher on ultrasound-mediated blood spinal cord opening, and Danay Valenzuela Rodriguez from Venezuela on cardiac ultrasound from MO to artificial intelligence in clinical routine. And this webinar will be moderated by Elisa Conofagu from Columbia University. So connection details will be announced through the UFFC website and social media channel. So again, thank you everyone for joining, and hope you see you in the next webinar.
Thank you very much.
Well, thank you so much.
Thanks a lot.
Thank you.